The topic for today's discussion is DNA replication. DNA is self-replicating structure and DNA replicates semi-conservatively. DNA replication is catalyzed by a set of enzymes. In the process of DNA replication, the DNA makes multiple copies of itself. It is a biological polymerization which proceeds in the sequence of initiation, elongation, and termination. It's an enzyme catalyzed reaction, and DNA polymerase is the main enzyme in replication process. So these are the general features of replication. Firstly, it's a template dependent process. The two DNA strands separate and each acts as a template for the formation of the new strand. And polymerization is a reaction which is dictated by a template strand according to the base pairing rules, where the adenine is present in the template, then the thymine gets added in the new strand, and guanine pairs with cytosine. The other feature is primer dependent. DNA polymerase needs primer to synthesize new strand on it. Primer is a small strand or small segment which is complementary to the template. It has a free 3-OH end to which a new nucleotide can be added. In this way, part of the new strand is already in place. Primer is hydrogen bonded to the template to form the primer template junction. Primer is a short nucleotide strand, which is also called as oligonucleotide. The primer for both the new strands is RNA primer. The RNA primer is synthesized by the enzyme primase, and the DNA polymerase moves along the template, adding nucleotides. Thus, DNA is synthesized by extending the 3' end of the primer. Afterwards, primer is removed by the DNA polymerase 1 and RNase H enzymes. Leading strand requires only a single primer, whereas on the other hand, the discontinuous synthesis of the lagging strand requires primer for each Okazaki fragments. For synthesis of lagging strand, hundreds of Okazaki fragments with associated RNA primers are required. So this is a picture depicting how the RNA primer is attaching to the template strand and how the primase enzyme is attaching the primer to the template. And the DNA polymerase keeps adding the nucleotides to the RNA primer, which extends and starts the replication process. The other general features of replication are it's a bidirectional replication. In most organisms, replication is said to be bidirectional. Replication begins at an origin and proceeds in both directions away from it, as shown in the diagram. Thus, in most organisms, two replication forks move away from each origin in opposite directions. It is also a semi-conservative mode of replication, where the Watson and Crick model suggested that DNA replication is semi-conservative, and it implies that half of the DNA is conserved. Only one strand of DNA is synthesized, and the other strand is the original DNA strand, which is the template, and that is retained. Each parental DNA strand serves as a template for one new complementary strand. The new strand is hydrogen bonded to its parental template strand and forms a double helix. Each of these strands to the double helix contains one single parental strand, which is the original parental strand, and a newly formed strand, which is the synthesized strand after replication. It is also semi-discontinuous. That means if the synthesis proceeds in the 5-3- direction, only one strand can be synthesized in the correct 5-3- direction. As the strands are anti-parallel, the other strand will have to be synthesized in the 3-5- direction. This constraint is overcome in an ingenious way. As the two strands unwind and the replication fork grows, one strand is synthesized in the 5-3- direction correctly in a continuous manner. This strand is called the leading strand. It grows in the same direction as that of the replication fork. Whereas the other strand is called the lagging strand that also grows in 5-3 direction, which is opposite in the direction of the replication fork. In the lagging strand, the DNA synthesis does not occur continuously, but in small fragments, which is called the Okazaki fragments. Later, these fragments are joined together and they are sealed with the help of the enzyme DNA ligase to form a continuous strand. So this picture shows you the three modes of replication, which is the conservative replication, the semi-conservative replication, as well as the dispersive model of replication. In the dispersive model of replication, you can see that both the copies of DNA have double-stranded segments of parental DNA, and the newly synthesized DNA is interspread. The role of enzymes in DNA replication is very important because DNA replication is highly enzyme dependent process, and the major enzymes which are involved are DNA polymerase, where the DNA polymerase there are different types. DNA polymerase 1, it's mainly a DNA repair enzyme, and it's involved in the 5-3 polymerase activity, 5-3 exonuclease activity, as well as 3-5 exonuclease activity. DNA polymerase 2 is responsible for primer extension and proofreading. DNA polymerase 3 is responsible for the in vivo DNA replication. The other main enzymes are helicase, which is the enzyme which unzips the DNA strands by breaking the hydrogen bonds between them. Thus, it helps in the formation of replication fork. Ligase is the enzyme which joins together the Okazaki fragments of the discontinuous DNA strands. Primase is the enzyme which synthesizes the RNA primer complementary to the DNA template. Endonucleases, these produce a single-stranded or a double-stranded cut in the DNA molecule. Single-stranded binding proteins are those which are responsible for protecting the single-stranded DNA into forming the secondary structures. Thank you.